This is our third and final mini lecture on neurons. And we're going to focus on the dendrites. Now you'll recall the dendrites have these projections of the cell membrane, and upon those projections is a receiving of signals. Those signals can come from sensory organs or perhaps other neurons. What is special about the dendrites? Okay, the dendrites have very different membrane properties from the axon. And those differences endow the dendrites with some properties that contrast the axon. The first property is the ability to produce depolarizations, hyperpolarizations, in other words, electrical currents from a variety of inputs. Okay, so they're effectively transducing a different kind of stimulus, and we're going to consider a stimulus that comes in the form of a chemical. They can take that chemical stimulus and produce electrical current. So we're going to consider how that's possible. Next, the dendrites include, or it's really the juncture between the dendrites and the axon, but in that region is where the neuron is most intelligent. That's the region where decisions are made. And the decision is about whether or not to generate an action potential that's propagated through the axon. So we're going to consider how that works. And then finally, once we understand how the neuron has this ability to make decisions, then we're going to see how the dendrites can integrate signals from multiple sources to inform that decision making that occurs. All right, so let's start the consideration of these properties by focusing on the interactions between two neurons. And in particular, we're going to look at chemical synapses. So chemical synapses allow for the release of a neurotransmitter from a presynaptic cell. So this is the axon of one neuron. And here's its terminal. That's the axon terminal. And then we have a dendrite here, which receives signals from that presynaptic cell. So we would call this the postsynaptic cell because it's below the synapse right here. The chemical synapse is an anatomical term. It's the juncture between the pre and postsynaptic cells. So let's focus in on that little space. Here up above, we have the presynaptic cell, and then down below is the postsynaptic cell. So in our consideration here of the properties of dendrites, we're actually going to start by finishing up the last property of the axons, because the axons are the part of the cell that releases the neurotransmitter. And so there are a few steps to this action of releasing the neurotransmitter. First of all, at the axon terminal, we have a high density of voltage-gated channels, but these are calcium voltage-gated channels. Calcium depolarizes the membrane, but calcium also can function as a signaling molecule. We're going to see this a lot when we talk about muscles. In this case, calcium is activating motor proteins within the cytoskeleton of the axon. Okay, motor proteins. Why motor proteins? Well, those motor proteins are associated with synaptic vesicles. These are little packages of neurotransmitter around the axon terminal. In this case, it's acetylcholine. That doesn't really matter so much. It's a neurotransmitter. It's a chemical that's capable of activating the postsynaptic cell. So let's consider the steps of the release of the neurotransmitter. First of all, we have the action potential reaching the terminal. Okay, the action potential arrives just the same way that as we've detailed in prior lectures about depolarizing the membrane. That depolarization is driven by voltage-gated sodium channels. Okay, so that depolarization arrives. That's the action potential. That's going to trigger the voltage-gated calcium channels, which allows for this influx of calcium in high concentration. That calcium is functioning as a signaling molecule which activates the motor proteins associated with the vesicles. Here we call them regulatory proteins because they're regulating the activity of exocytosis. But you can just imagine a little protein that walks its way along the microtubules to move this vesicle towards a synaptic cleft down here. Eventually, it will dock with the membrane that's facilitated by a protein, and that's going to trigger the release of our neurotransmitter. The neurotransmitter is released, of course, because it's a chemical. Chemicals will move according to chemical diffusion. We have a low concentration in the cleft, and therefore the acetylcholine floods into the cleft because it has that very low concentration. So let's pick up from that point to consider what occurs in the postsynaptic cell. So we're going to move a little bit here. So we're focused in the center here on the cleft, the extracellular space between the pre and postsynaptic cells. That's the space that we have here. And at this point in time, we have a high concentration of our neurotransmitter. And what we can see down here is that one of those molecules of the neurotransmitter has been bound to this what we call ligand-gated sodium channel. Now, ligand-gated because it binds to a chemical in order to change its conformation and allow permeability. In this case, it's permeability for sodium. Sodium is the ion that is used for an excitatory chemical synapse. So we have different types of chemical synapses. The ones that involve sodium, because of that very positive equilibrium potential for that ion, the enhancement in permeability that emerges because of binding to that neurotransmitter is going to drive that depolarization. That's what makes it an excitatory synapse. Okay, so we get a conformation change. Sodium moves down its chemical concentration gradient. That's going to drive a depolarization of the membrane on the postsynaptic neuron. So this is the region of the dendrites right around the chemical synapse. Note here, this is not an action potential. You do not get action potentials in the dendrites. Action potentials require voltage-gated channels, and those do not exist in the dendrites. Instead, you're going to get a graded potential. Okay, anything that's not an action potential is a graded potential. And so that graded potential in the case of an excitatory synapse is going to be in a more positive direction from the resting membrane potential. That's what we call a depolarization. Not all chemical synapses are excitatory. So here we're looking at a different site. Okay, chemical synapses are of a particular type. So now we're looking at an inhibitory chemical synapse, which is at a different site on the dendrites. Okay, this is the same postsynaptic cell, but it can receive both excitatory and inhibitory synapses. Here, this ligand-gated channel is specific to chloride. So chloride ions will be permitted to move across the membrane when they bind to a neurotransmitter. Chloride has a very negative membrane potential. It's similar to potassium in that respect, even though the high concentration is on the outside of the cell. But because of its negative valence, when the permeability is high for chloride, it tends to hyperpolarize the membrane. Okay, and a hyperpolarization is what we consider to be an inhibitory action. All right, and so here we have the chloride moving across the membrane. A little reminder here that chloride has that very negative equilibrium potential. And so that's going to make the membrane more negative. So instead of a depolarization, we're getting a hyperpolarization. There are other types of synapses. So here we have an example of a postsynaptic membrane or a chemical synapse that okay, so we're considering a different site here. And for this chemical synapse, instead of being excitatory or inhibitory, we instead have what's called a G protein coupled receptor. So our neurotransmitter in this case is again binding to a transmembrane protein, but the response to that is not to directly affect ion permeability. Instead, we're activating a variety of enzymes and maybe transcription factors that are going to influence the behavior of the postsynaptic cell over generally a longer period of time. Now, these can be a large class of second messenger pathways are a large class of pathways that can alter the state of cells, including long-term effects on their membrane potential. So, for example, they can affect the density of ion channels and also their open and closed state. They can trigger transcription of that's going to result in a higher density of leak channels of the membrane. So you can think of second messenger pathways as a means of modulating the membrane properties of the dendrites. They can also have other effects on the cell as well. But if we just focus on their effects on the permeability of ions, a G protein coupled receptor can result in a slight hyperpolarization of the membrane, making it less likely that an excitatory synapse is going to trigger an action potential in the axon, or they can make it more likely. They can slightly depolarize the membrane, but still have that depolarization be below threshold, so that when an excitatory synapse arrives, it makes it more likely that an action potential will be generated. So we're not going to go into lots of detail on all of the variety of second messenger pathways, but we're going to see throughout this course that they will make these occasional appearances. Okay, so we have just within chemical synapses this illustration of three different ways that we're transducing a chemical source of energy into its effects on electrical current across the membrane. Next is this key property of decision making that is exhibited by neurons. This ability to decide whether to transmit signals. And then after we explain that, then we're going to consider the integration that occurs across the dendrites. All right, so generating an action potential. Let's consider an excitatory synapse. And we now see that the action potential from the presynaptic cell can result in this diffusion of sodium into the cell at the excitatory synapse. The key, decision-making algorithm for the cell is if that depolarization can manage to trigger the voltage-gated sodium channels in the axon, then that's going to cause an action potential. So what we have here is a site where decisions are made. And that site is only special because it's where the axon begins. So it's really this juncture between the axon and the membrane that covers the dendrites. This region tends to be sort of a bottleneck of the cell. And so this anatomical term of the axon hillock refers to the shape there. But functionally, this is referred to as the spike initiating zone. Spikes are a term for action potentials. So this is where the action potentials begin. It's at the base of the axon. And that is indeed the site of the start of the action potentials. So let's perform some virtual experiments on the neurons to understand how this all works. What we're going to do is monitor the membrane potential. We're going to start off by monitoring right at the site of the chemical synapse. And so when an excitatory signal arrives, that results in a depolarization. This is a graded potential, kind of looks like an action potential, but it's not because, of course, there are no voltage-gated channels of any sort, including sodium, in the dendrites. Now, since this is a graded potential, just like we saw what occurs in a myelinated axon, you're going to get a degradation of that excitatory stimulus as you move away from the site of the chemical synapse. So let's move a little bit along the dendrite here, and we see indeed we have a drop here in the intensity of that depolarization, and there's a little delay here, which is reflecting the time that it takes for chemical diffusion to work. If we look just above the axon hillock, we see again this depolarization, it's less intense. But if that depolarization is above threshold, the magic number of minus 55 millivolts, it's going to trigger the voltage-gated sodium channels in the axon and boom, result in an action potential in the axon. So we have a series of graded potentials in the dendrites. They're strong enough to activate the voltage-gated sodium channels, which results in this action potential that we see in the axon right here. Again, the rule here is that if the action potential creates a membrane potential above threshold at the axon hillock, then you're going to get an action potential. And we see that at this site really close to the hillock, it's very strong. It's well above minus 55 millivolts, resulting in an action potential. Okay, so that's how decisions are made. How are signals integrated by the dendrites? Well, the simplest case is what we see here. You get an excitatory synapse. If it's strong enough, you get an action potential. Pretty simple. Where it starts to get more complicated and more interesting is when you get a variety of signals. So you can have multiple signals coming from a single chemical synapse. That would be an example of what we call temporal summation, which is a rapid succession of action potentials that affect the membrane potential at the axon hillock, where decisions are made about whether or not to generate an action potential. So in this example here, we're going to look right at the site of the chemical synapse. We give it two quick depolarizations that are coming from two action potentials from the presynaptic cell. So right here is where we're monitoring these two depolarizations. As we track the propagation of that excitatory signal, we see that it does decrease in intensity. We have a delay in time. But actually, one thing, if I sort of back up here real quick, one thing I want to point out here is that it's possible to get a second depolarization. This is unlike what we saw with the refractory periods in the axon, because those two action potentials are equal in intensity. You don't have action potentials varying in intensity. And same thing on the postsynaptic cell, on the dendrites there, they receive that signal in equal intensity. But on the postsynaptic side, we see that the second stimulus results in a greater depolarization than we had in the first one. Why is that? Well, graded potentials are operating in this case with a slower time scale. That is, the sodium that drove the initial depolarization, some of it still remains. We see that we haven't gone back to the resting membrane potential at the time that the second excitation occurs. So what we have is a summation of signals. We're building on the first signal to create a more intense second signal. So this is possible in the dendrites. It is not possible in the axon. You don't have a summation of signals in the axon. Okay, you either have an action potential or you don't. And when you don't, you're back at the resting membrane potential. Or you might be in the relative refractory period, in which case that's going to hurt, not help your ability to create another depolarization. So you can see that just by pretty subtle differences in the composition of the membrane and the dendrites, you have very different properties. All right, so that second really strong depolarization is more likely than the first to create an action potential. And as we track that excitation through the cell, we see that it does degrade a bit and it's delayed in time, but if it's above threshold, by the time we get to the axon, it's going to create that action potential, which is what we see occurred here. All right, so that's temporal summation, the ability to create a stronger stimulus with repeated activations of an excitatory stimulus. Now, you can also do this in an inhibitory sense. If you had an inhibitory chemical synapse that electric chloride across the membrane, that if you have repeated action potentials at that site, then you make it less and less likely that you're going to get an action potential in the axon. All right, spatial summation. Spatial summation occurs if you have multiple presynaptic cells. So let's add another presynaptic cell. We're going to move our electrode there so we can monitor what emerges from that second chemical synapse. So here we have the first chemical synapse. It's a rather small excitatory signal. And very close in time, we're going to get a second one at that second site. Both sites allow for an influx of sodium. So right here in red, we have the sodium influx. So that's going to increase the amount of sodium that moves and diffuses into the interior of the cell. It's another means of summation, not temporal here, but because we have two sites working in concert, it qualifies as spatial summation. So what you get down here, really close to the axon hillock, is an even greater depolarization. So even though we would expect individually those two depolarizations to degrade with space because they both occur similar in time, then they sum and create an even greater depolarization. And we get an action potential because that's above threshold. Now let's consider another example here where we have a really small depolarization. Our second depolarization slightly more delayed this time, and it's also pretty small, below threshold, so that when we sum the two, we've got a stimulus that is below threshold. This maybe should be drawn a little bit differently because it doesn't reflect the summation that could occur. But it's meant to illustrate the fact that not all synapses create an action potential. If it's below threshold, then of course it won't do that. Now, summation can also reconcile the inputs from both excitatory and inhibitory synapses. So in this example, we were considering only two excitatory synapses. Here, we're going to add an inhibitory synapse. So the way that that looks is here we have first the excitatory one, but now we're going to actually hyperpolarize the membrane at this second site. Okay, so imagine chloride driving a hyperpolarization here. That inhibitory synapse can essentially block the ability of the excitatory synapse to create an action potential in the axon. And that's exactly what we see. Here's the summed signals real close to the axon hillock. And then finally, we get nothing in the axon because that inhibitory signal has blocked the excitatory one. All right, so this is just illustrating some of the real fundamental properties of integration. It gets much more complex when you consider networks of neurons. So I'm just going to illustrate a couple of examples. We're going to see lots later on. Here we have multiple presynaptic neurons influencing a single postsynaptic neuron. They're going to be, that postsynaptic neuron is integrating signals from the three presynaptic ones. In this case, we've got two excitatory chemical synapses, and then the third is an inhibitory chemical synapse. So now this single cell is weighing the relative intensity of these excitatory synapses against an inhibitory synapse. And in this particular case, the excitatory synapses win, but that won't always be the case, quite obviously. So we have the ability to facilitate the integration of signals from multiple sources. On the other hand, it's possible to amplify signals. So here we have the reverse, one presynaptic neuron that has an axon branching to multiple postsynaptic neurons. And imagine that each of those postsynaptic neurons are innervating what we call an effector organ, which is sort of a general term. This could be a gland, or each of these could be muscle fibers. So when you have one excitatory signal, if that signal is passed on and serves to excite all of those postsynaptic neurons, then you have the ability to activate lots of effector organs. This could be lots of muscle fibers to generate a lot of force, lots of glands to secrete a hormone. So networks can also amplify signals from either multiple or single neurons to lots of effector organs. So what's so special about the dendrites? Well, they do have this ability to transduce information into electrical current. We've seen that both with excitatory and inhibitory chemical synapses. At the juncture between the dendrite membrane and the axon membrane, we have the spike initiating zone, which is where the site, it's the site of decision making that occurs in the neuron, whether or not to transmit an action potential. And because of these properties, dendrites have the ability to integrate multiple signals, both through spatial and temporal integration.